Hello and welcome to this webinar, Broadening the Horizon of Drug Discovery, Use of Unperturbed Native State Cells to Uncover True Biological Targets at Single Cell Level. I'm Lou Rahman, Publishing Director at DDW. Today's webinar will be presented by Dr. Camillo Egidio, Senior Director, Application Development, Levitas Bio. Extensive manipulation, mechanical force and labelling methods used to enrich cells activate transcriptional programs that mask molecular profiles truly significant to any underlying disease. The consequence is the identification of drug targets that are not relevant to the biological process under study. Levitas Bio will present how single-cell RNA sequencing has progressed the field of drug discovery and how upstream sample preparation methods that preserve cells in their native state is critical in identifying relevant biomarkers for drug discovery and development. In this webinar, you will learn about the relevance of single-cell RNA sequencing in your drug discovery toolkit. You will find out about obstacles that stand in the way of precise molecular signatures and lead to misguided biomarker identification, and you will also learn how to use magnetic separation of viable cells present in low-quality samples for downstream biomarker discovery. First, some background on Camilla. She's a molecular biology scientist with experience in developing new products and systems to support biomarker discovery and patient testing in the biotechnology industry. In her current role, Camilla and her team reveal how magnetic levitation can segregate finite viable cells present in low-quality samples while preserving intact transcriptional signatures for accurate molecular characterization and biological discoveries. Okay, Camilla, I'll hand over to you now for the presentation.
Well, hello, everybody. My name is Camila Egidio, and I'm a Senior Director of Applications Development at Levitas Bio. And today I'm going to talk to you about broadening drug discovery horizons and using unperturbed native state cells to uncover true biological targets at single cell level. And my talk is in the context of the drug discovery being a long, expensive process with a really low success rate. And to enable a breakthrough in this field, what really needs to be done is to look at the high failure rates and what's happening in there, which really clinical efficacy is at the core of that issue. And for us to allow improvement in clinical efficacy, we need better and improved biomarkers. In an area where we just completed the human genome and transcriptome, the telomere to telomere consortium just finalized the 8% heterochromatin gap that was missing from the initial human genome project. We see very little increment in new novel elements that are protein coding. So it's highly unlikely researchers are going to find complete new and novel biomarkers for association with disease. So what we really need is to really understand how the biomarkers we have found so far behave and their role in specific disease and their subtypes. So take, for example, what has been done by the Genome Atlas Network in 2012, comparing all of the omics data from breast cancer to several different subtypes of cancer disease. And what they found is that there was a huge molecular heterogeneity within each one of the subtypes of breast cancer. And they found that the basal subtype had a very similar molecular profile as the ovary serous subtype of cancer. And that allows shared opportunities of therapeutic selection for the breast cancer tumors that weren't really in the picture before. And that really improves and progress drug biomarker discovery, because now you can use drugs that might have been already developed for ovary cancer and that are in a much more improved and progressed state of development for a novel disease. So we have now an advancement in technology that really has empowered a lot of stratification studies at the single cell level, and that has only improved additional subtyping and biomarker selection. So you see here, these are 3 papers from this year, so very hot from the press, where there is a huge number of single cells analyzed. And these were all done in a very unbiased way to really interrogate the full landscape and the cellular component of this human disease. So hepatocellular carcinoma, 71,000 single cells were analyzed, and 7 new molecular subtypes were found that are associated with disease prognosis. And then in biliary tract cancer, you have over 45,000 single cells analyzed that have highlighted use of markers that can enhance immunotherapy strategies. And in gastric cancer, over 200,000 single cells were analyzed. And that has also provided the discovery of a novel and rare cell subtype that's characterized now a cancer subtype that's associated with poor clinical prognosis. So this is the type of research that will really lead the way and offer breakthrough progress in drug discovery. And this has only been enabled by the improvement of technology. As you can see here in this sketch, there is a huge number of now flavors of RNA sequencing technologies. And we've seen improvement in the library preparation side with different elements that improved parallelized high throughput data generation. So now you don't just include several different samples in the same run, but you can also evaluate different markers, whereas protein, RNA, and DNA all together in the same run. Additionally, we've had a lot of improvement in the sequencing side too, with third generation sequencing that has allowed improved accuracy and also turnaround time, and particularly cost. What we haven't seen is really a lot of technological advancements in the upstream single cell preparation part. And that is a highly manual process that contributes to the greater source of unwanted technical variability in downstream single cell sequencing. The worst part of that is that it actually, because of the highly manual character of these steps, is that it does affect the gene expression profiles in the cells. Cells might get from a native state to activated, and now their transcription profile is completely different. And that confounds all of the biological conclusions one can take from a human disease and make conclusions around best biomarker to be used for that disease. So with all of this different complex sample types that needs to be analyzed for single cell RNA sequencing, the big bottleneck that we really see is in the sample processing side. And like I mentioned, the manual workflow and high variability also added to the low cell viability of these really challenging cell subtypes. High percentage of debris and cell death creates these altered gene expression profiles. So for example, in cryopreserved cells, Schaefer and collaborators in 2018 evaluated 77 different PBMCs samples that are stored in retrospective biobanks, which is a huge opportunity to unlock a database that can be used for clinical trials. And what they saw is that after these cells are thawed, we have a variability in the viability between 20 to 85%, which is really adding this variability downstream to the single cell RNA sequencing. Whereas compared to healthy donors or samples that were never frozen, you have a much higher viability and consistent performance on that metric. But we can't afford to always analyze fresh samples. Difficult samples will always exist, and we owe it to patients that donate their material for science to be able to unlock significant biological data from these samples. So what we really need is a breakthrough in technology to empower that. And here I'm not going to really talk about the technology yet, but I'm going to show you what it can do. So in this video, we have labeled a cell suspension that have really low viability, around 2%, and they are labeled, the live cells are in green and dead cells are in red. So what you see is green cells or live cells levitating to the top of this levitation chamber, and the dead cells are at the bottom. So you do see a few green cells that are maybe intermingled with the red cells. These are live cells that sometimes do get stuck in the debris in the dead cells. But you can see how clean the top fraction is of what we call the viable fraction of the cells that are being levitated in the levitation chamber. So the stains are not required or necessary. These were used in purpose for demonstration only. And then what happens is that after the cells have levitated for a number of minutes, in this case was 20 minutes, which is pretty standard, we harvest this material using different pressure on the top and on the bottom. And these two, they output in two different separate chambers in a cartridge where they can now be collected and used for downstream sample processing and data generation. So what you saw in the former slide, in the video, was actually an increase in improvement in viability from 2% to 80%. So we started with 1 million cells, and here on the y-axis on the left, you can see the total number of cells that were input into the levitation cartridge. And we started with 1 million cells, and of those, we had about 20,000 cells that were viable, or 2% total viability. And then after the 20-minute levitation workflow, we recovered 11,000 cells that are sufficient for downstream RNA sequencing analysis at 8% viability. So that means you have no debris, no dead cells in your suspension that now you can use effectively for RNA sequencing. And this workflow is very consistent and reproducible. Here you see a number of cells, samples, in here on this graph showing viability on the y-axis, and a number, each dot represents a sample tested. And you have here about 12 different samples at different inputs, ranging from 10,000 total cells to 1 million. These were cryopreserved PBMCs. And after that, you have a 5-fold enrichment of viable cells in average. And this is a method that uses no labeling, and it's hands-free and automated. So user can just step away during the 20-minute run. And this workflow can be used in a huge number and a wide variety of different cells and applications. So here, in the context of drug discovery, with those CRISPR-edited cells that you want to make into models for your disease or biomarker selection, and evaluate how these drug candidates might affect cells downstream in the process, you have CRISPR transduced cells that are at 8% viability before being added to the levitation workflow, and then after that, we achieve 71% viability. And that now is amenable to higher and better quality for downstream data. And here, another example of what can be done for drug discovery is that in a PDX model, the cells that were levitated achieved a much higher efficiency compared to cells that are not levitated. So in this example, in the laboratory at UCSF from Ernesto Diaz-Flores, what they've done is to obtain cryopreserved leukemic clones, thaw them and resuspend, and then a subfraction of those cells were subjugated to the levitation technology workflow for viability enrichment, and a subset was just not going through that process. And then these cells were now, a number of them, used for xenotransplant to reconstruct the leukemia model in mice. And in one example, we only use 100,000 cells from the levitation workflow with 95% viability. And in the other arm, we had cells at mixed variability, and 1 to 2 million cells were used to reconstruct the model in mice. And the result of that is that even after using 10 to 20x higher number of cells for the transplantation, there was still a lag of 5 weeks. So it took 10 weeks, 10 total weeks for the model to be reconstructed in mouse, as compared to cells that were levitated, which only took 5 weeks to fully reconstruct the disease model in mice. Okay, so in that sense, I want to walk you through now about how the levitation technology can really improve the data that you get downstream and what's actually happening there that allows this improved results in different applications. So in one of them, we focus on mouse brain cell subtype characterization. And for that, what we use is a cold brain cell dissociation kit that's a proprietary from Levitas Bio. And we did a step to remove red blood cells and myelin because this brain tissue was not perfused. Then we did a 2-arm study in which we had a levitation technology used in one subset and standard technology in another one. And those 2 different cell suspensions were now used for single-cell transcriptome comparison between the 2 different experiments. The goals for this study was to evaluate the performance of the viability enrichment between the different methods and understand how these unbiased methods for viability enrichment can impact the gene expression profile of the cells. So here is just a sketch of the experimental design, the non-perfused mouse brain that was used, the LeviPrep mouse tissue dissociation kit, which takes about 60 minutes, and it's based on mechanical and enzymatic processes to do the single cell suspension generation. And then the myelin and RBC removal, they're just standard techniques that are used and takes around 50 minutes. And then a subset of the cells were taken through the levitation technology for 20 minutes without any labeling, just for unbiased enrichment, whereas the standard method was used for viability enrichment. And now you have the droplet single cell library preparation in RNA sequencing. So one of the results that we obtained from this was that the LeviPrep cold dissociation method can retain all of the resident cell subtypes from brain. As you can see here, from the full data set generated and not parsed into the different viability enrichment methods, we can see the astrocytes, microglial, endothelial, neuronal, and oligodendrocytes all represented in the data set. And here is just showing the biomarkers used to identify each one of the cell subtypes and the composition of the cell subtypes in this data set. So this was a really valuable result to show that LeviPrep can indeed separate all of these very sensitive cell subtypes from brain without losing representation of the main cell subtypes. And then what we zoomed in the microglial compartment, and that's because this is a typical, really important cell subtype in the brain that's under study for neurodegenerative diseases and a model for a target for PDX development and drug biomarkers. So what we identified was 4 different microglial cell states that were characterized by their different expression profiles. So you have the majority here being resting state, and then you have a subset that is proliferating, a subset that expresses phagocytic activity and inflammatory. And then what we did was now identify that first around 10% of these total microglial cells detected were in activated states, and that's highlighted here in purple. And what we did was to now separate this full data set into the cells that were generated by levitation technology and cells that were enriched in viability using standard method. And what we saw, to our surprise, was that we actually had cells in the levitation technology subset that we had twice less number of cells activated present in the levitation technology workflow as compared to the standard method, which means the standard method subjugating the cells to more stress that creates activation profiles that really affect the downstream gene expression of these cells. And in this graph here, you see that cells in resting state were over 95% in the levitation technology as compared to standard methodology, which is at around 86%. And here, the increased in percentage of cells detected with standard methodology that are in the activated states. Well, to corroborate this result, we actually looked at standard native state microglial cell markers, and we actually found that between the 2 different methodologies, with the standard method, we had similar gene expression as found in the levitation technology as expected, because these are native or resting state biomarkers of microglial cells. And then comparatively, when we look at those biomarkers of microglial activation, we do see up to 6 times higher expression of those markers in the standard methodology. And this really just underscores how the levitation technology is a gentle method that preserves cells in the native state, in which should be the best state to characterize the cells for disease understanding and characterization. Okay, so I'm going to move on into our second case study, which really talks about the dissociation of tumor cells from sarcomatoid carcinoma. And this is a rare lung cancer subtype that's typically associated with poor prognosis. So what we did is to work with DTCs from this cancer type, and we had 3 different arms for our study. 2 of them using levitation technology, in which we provided just the workflow for viability enrichment, and a second workflow in which we also did CD45 positive cell depletion, which is pretty much removing all of those immune cells from the tumor microenvironment to only analyze tumor-related cells. And the third arm was no enrichment, no viability enrichment, and no CD45 cell depletion. Then we performed single-cell transcriptome comparison of about 4,500 cells across all of the 3 methods. And the goals of this study was to evaluate the performance of the viability enrichment with and without CD45 cell depletion, and also to review the downstream technical impact of the levitation technology workflow on RNA sequencing metrics. So here, summarizing further the experimental design for the DTC TOS. So these cells are actually purchased from Discovery Life Science Lab, and we obtained them at 2 million per vial. These were cryopreserved, and once they arrived in our laboratory, we thawed them according to the vendor guidelines. And in terms of viability, prior to cryopreservation, Discovery Life Science stated that they were at 83% viability, and then after they were thawed, we made the same assessment in our lab, and we saw that there was a 39% viability. So just showing that drop in viability just from the cryopreservation method on its own. So then we subjugated the cells to the Levi-cell only or the viability enrichment workflow. And also a subset of those were subjugated to the Levi-select with the CD45 positive depletion and viability enrichment, and then the no enrichment workflow. All 3 of them had the same number of cells input, and the output was used for, again, the cell library preparation RNA sequencing, and we used 5,000 cells from each one of these conditions to do the library preparation. So I think that really interesting result we obtained at first on the sequencing data was that there were a major significant differences between just the levitation technology and the cells obtained from no enrichment. So just looking at those 2 separate workflows first, we had on average a much higher number of cells sequenced with the levitation technology compared to no enrichment. And this is parsed by the 2 different methods, right? We didn't sum both of those 5,000 cells as input. We only got one of those conditions to compare to no enrichment. And this is the difference that we saw. A lot more cells were captured when we used a suspension that was an output from the levitation technology compared to no enrichment. And this means that we actually recovered a much higher number of cells from the input of 5,000 at around 68% with technology enrichment compared to 23 with no enrichment. As expected, the number of cells detected with levitation technology was lower because we added these cells to the same sequencing run. So we obtained higher number of reads for no enrichment. But even though we got that different number of reads or throughput coverage or depth, we did identify a number of transcripts much higher across the cells using levitation technology compared to no enrichment. And of course, we had higher number of genes detected per cell, and a higher percentage of those reads mapping to the transcriptome, showing that the amplification of the library was much more efficient with levitation technology compared to no enrichment. As a consequence, we had a much higher number of genes detected in this workflow compared to this one. And this is really to show how magnetic levitated cells, they produce a transcriptome information that's much more in depth than non-levitated cells. So you're getting more information from the same cell subset compared to no levitation enrichment. And the reason for that is really because when we use the DTCs with no enrichment, we had 39% viability compared to the cells that we use from the levitation technology at 87% viability. So it's a very simple message. You have low quality cells coming in, you're going to have lower quality sequencing results as well. So when you enrich your cell suspension for viable cells that are happy and have intact membrane, you're now enriching your data set for more biological information that's relevant to your research. So this can be shown also from the cDNA profiles of the libraries generated, in which the levitation technology generates a cDNA library that has a very clear amplification profile as compared to no levitation, in which you might have a lot of droplets that capture just debris in dead cells and are not really generating signal and contributing to the sequencing reads for that library. Another interesting result that we found is that the unenriched DTC cell suspension generates less data using droplet single-cell library preparation. And this is pretty much in line with the other result, which shows that using levitation technology, we detect a higher number of cells across all these relevant subsets for the disease. And this is shown by over threefold representation of cancer cells in the data set. So it's important again to highlight it's the same number of cells that went through both workflows for library preparation, right? So we're capturing a lot more cells from those really rare and important samples from patients compared to a workflow that does not do any viability enrichment. And what we see here is that when we do the negative selection of DTCs, which means we are positively selecting for the CD45 positive cells, we have in a workflow that we didn't do the enrichment for DTCs, we collected 1,400 immune cells. So data on all of these cells was collected from the sequencing results. Whereas when we excluded these cells in the same LeviCell workflow, we had an enrichment of 99.9%. And we only got one B cell in this workflow, depleting the CD45 positive cells, which really shows how we increase the amount of data that we collect from the same workflow. So now going from 43 epithelial cells collected in a workflow that we had no viability enrichment, we enriched this data set to 269 single cells collected for this cell subtype that's very relevant. That's pretty much where the disease originated from. So we have a 6 times greater representation of these rare, these important cancer cell subtypes when using LeviSelect and LeviCell. So in summary, I just wanted to go back to the fact that drug discovery success does depend on better biomarker selection. And the single-cell RNA sequencing is a powerful tool to support the improved biomarker identification by providing deep understanding of disease biology and molecular characterization of its subtypes. While we've had a lot of breakthrough improvements to allow deep atlasing studies, there is still challenges in the upstream sample preparation steps for the single-cell RNA sequencing workflow. And those include an inability to rescue low-quality samples, those samples that are in low cell number and viability, samples from biopsies, and retrospective biobanks, for example. And use of manual and cell activating steps produce data variability and introduce gene expression artifacts that might confound data and the biologically relevant conclusions that researchers can make on biomarker selection. So I hope that today I have shown to you that the levitation technology workflow can rescue low number of viable cells, up to 10,000 cells, for downstream single cell RNA sequencing. And that is unprecedented. There is no technology out there nowadays that work with this low level and number of cells. And it does so in an automated, hands-free way in a 20-minute runtime with a label-free workflow. And this technology, combined with the LeviPrep cold association method, produce single-cell suspensions from brain and other difficult tissue types, preserving the representation of all of those cell subtypes that are relevant in brain studies, and decrease cell stress and expression of activated markers by 6-fold. And when combined with the LeviSelect CD45 positive cell depletion method, levitation technology produced sequencing data on dissociated tumor cells that have superior performance, producing 45% of reads mapping to the transcriptome versus only 6% for non-enriched workflows, and maximizes the biologically relevant data with a cell recovery of 68%. So you collect 68% of those really rare and important cell subtypes versus 23 for non-enriched workflows. So thank you so much. I'd like to especially acknowledge the team generating all this data and the Levitas Bio team. Thank you.
Thank you for that presentation, Camilla. That was really informative. So before we go to questions, I'd just like to remind everyone that this webinar will be available to access on demand on the DDW website, www.ddw-online.com. Okay, Camilla, are you ready for the questions?
Yes, please.
Okay, and we'll start then. So the first one that we have is, how does levitation technology work? Will it damage or adversely affect my cells?
Right. So, well, it's not magic, I promise you that. So levitation technology is used through the use of a levitation agent that's spiked into a cell suspension. And that's what really much creates the levitation that you see of the live cells. So levitation agent is a solution that's attracted to magnets more strongly than the cells. And when I show that video and showing the levitation chamber, you have magnets on both sides of that chamber, and that's what creates the levitation profile of cells. Now, when you have all these three forces acting on it, you have cells with different physical properties, and the cells will levitate higher or a little lower depending on their physical properties. But all in all, what happens is when you have a suspension that has both live cells and dead cells, those dead cells take up on the solution from their environment. So what happens is that this lowers their levitation force, and they go into the bottom of the levitation chamber. Whereas live cells, they exclude the suspension in their environment, so they levitate higher. And then by applying pressure, very gentle differential pressure to harvest these two different suspensions inside of the levitation chamber, we are capable of harvesting this solution separated from the dead cells. So at the end, what the user collects in the levitation cartridge is two different suspensions, one with live cells and one with dead cells. And to answer the question about how it might affect your cells, so the levitation agent is actually the same that's used for the MRI contrast that we take up when we go and do this exam. So it does not affect the health of the cells. And it has been shown to be compatible with a huge number of sample types and cell subtypes. And there is also opportunities to improve, or even for each cell subtype that is under research, to tweak conditions on the cartridge to ensure that appropriate separation of down to 1% of difference in density between cell subtypes.
Okay, thank you for that answer, Camilla. So the next question that we have been asked is how to explain the faster reconstruction of the PDX model using levitated cells?
Well, that's a great question. When we evaluate the performance of the levitation technology, we usually use the AOPI staining, which is a common viability stain that's used to evaluate viability. And we do that before Levicell and after. But that is actually short selling the effect or the capability of the technology, because what we see, it's not just viability enrichment is one of the metrics that we use that's very easy to observe before and after. And they're very quick methodologies that can be applied into any lab. But what we actually see is that cells that go through levitation, they're happier. They're more highly functioning, so they can actually recapitulate these biological processes much faster than cells that are not levitated or they're just selected for viability using different methods, because they go through a lot of pressure or harsh mechanical conditions for that viability improvement. So what we see is faster cell culture, faster PDX model regeneration, and a number of other metrics that actually showcase how these cells are more active and they're doing their functions that they usually do when they're in a native state.
Okay, so in the brain study, how does the standard technology affect the downstream gene expression of the cells?
So what we see is that when we have a cell suspension that has mixed viability and we have a high number of debris or dead cells collected, the droplet-based library generation for single-cell RNA sequencing captures also some of that debris and dead cells. And what in the downstream step of actually going through the amplification steps, that material does not generate signal like was shown in that graph from the bioanalyzer. So we don't have specific amplification of the RNA in the cells just because either the cells are dead and that RNA has been now degraded, or because there is debris that was encapsulated by the droplet generation library prep. And by enriching the cell suspension in viable cells, you now have a much higher percentage of cells that are viable, that have the membrane completely intact. And that's what's going to generate higher amplification specificity of the RNA that's within those cells. And what you get at the end is more information for the same cell suspension that you put through for library preparation when you have levitated cells.
Okay, thank you. In the DTC study, how were the CD45 positive cells depleted from the suspension?
Yeah, so the DTC cells, they were subjugated to a step prior to the Levicell viability enrichment, in which in 5 minutes they were labeled with a CD45 antibody, and this antibody is attached to a magnetic label. So those cells, when they go now through the levitation chamber, there is not just selection for viable cells, and those are DTC cells. They're floating and levitating to the top of the chamber, whereas the CD45 positive cells, they are labeled with the magnetic label, and then they are attached to the bottom of the levitation chamber. And then when we harvest the DTC cell suspension, what we get at the end is the material without those CD45 labeled cells. So there is no labeling or manipulation of the DTCs that are targeted for the downstream study.
Lovely, thank you, Camilla. And the last question that we have is, can your instrument handle cells of different densities, and if so, how?
Yes, absolutely. So like I mentioned before, differences in up to 1% on the different cells can be resolved in the instrument, which means that you don't just separate cells based on viability. You could also have cells of different density in the same suspension, and you can also tweak the conditions in the instrument to separate these cells in the top and bottom fraction and harvest them in 2 different outlets in the cartridge.
Okay, thank you, Camilla. Those were great responses, and thanks again for your presentation.
Thank you.
If you'd like to listen to the webinar again, it will be available on demand at www.ddw-online.com. You can now view Drug Discovery World content online on your mobile or tablet or via the DDW app, which can be downloaded via the App Store or Google Play. Thank you for listening.